Okay. Okay. So we have actually drawn all the basic components needed to explain the interplay of proteins involved in intrinsic pathway. Now listen with all your attention. You know what is this? What is the name of this protein? Which is actually, this protein is actually inhibiting this black one. Okay. Now listen with all your attention. The black one here is actually this back and backs, and this protein which is inhibiting back and backs is actually BCL2. Why BCL2? Why BCL2 is actually inhibiting? Why BCL2 is actually inhibiting back and backs? There is a reason for it. Listen with all your attention. Back and backs is actually, when it is not triggered, they will actually form monomers and they will be embedded. Okay, they are transmembrane, proteins. They are, transmembrane proteins, and they are embedded in the mitochondrial outer surface or outer membrane. Now listen with all your attention. When proper signal comes for apoptosis, what will happen is these back and backs will come together and they will form a channel. They will form a channel, oligomeric channel. Okay, and through this channel cytochrome C can exit. Okay. Now remember with all your, now listen with all your attention and remember this for your entire life. When cytochrome C exits mitochondria, it is dangerous. We have already discussed why cytochrome C is present in the intermembrane space, right? In our electron transport system class. Now listen. When cytochrome C actually exits the mitochondria, then via many different pathways, obviously we will discuss in great detail the pathways, caspase 3 is activated. Okay, that's it. And you know what happens when caspase 3 is activated? The cell dies. Now listen. Why cell dies? Because the function of caspase 3 is, because caspase 3 acts like an executioner caspase, and the function of caspase 3 is now listen with all your attention. What is the function of this BCL2 or BCLXL? Okay. The function of BCLXL or BCL2 is same, that is, it actually inhibits the formation of this, protein channel. This BCL2 or BCLXL actually inhibits back and backs, and therefore this protein channel is not formed in a normal mitochondria. But when this mitochondria is triggered, when this cell is triggered, when the cell is under stress, this BCL2 or BCLXL inhibition of back and backs will be removed, and they will unite and they will form a oligomeric protein unit, and this protein unit will act like a protein channel, and through this protein channel cytochrome C will go out into the cytoplasm, and through multiple pathways we will all discuss these pathways. Through multiple pathways caspase 3 will be activated and death of the cell will occur. Okay. Now listen with all your attention. It's very important point to understand here that in this discussion only we have already discussed anti-apoptotic proteins, that is, and we have already discussed the proteins which forms the channel, right, that is back and backs. Now when we will discuss these class of pro-apoptotic proteins which inhibit anti-apoptotic proteins, this class of proteins are puma, bid and bam. We will discuss later when we will be talking about the examples. Okay, in neurons these things occur. Okay. Now listen with all your attention again. So this is the basic, thing about the protein, interplay. Okay. Now we'll discuss in great details the pathway, actual pathway of intrinsic, actual intrinsic pathway of apoptosis. Then we will talk about the examples in C. elegans specifically. Okay. So oh, one thing I have forgot to tell you people, listen. There is very important, information regarding these proteins. I have forgot to, mention that. It's a very, lucid information, very easy information, but it's very useful. Okay. You know that proteins involved in apoptosis are known by the name of BH1 to 4 domain proteins. Why? Because I am drawing these 4, now listen. When I am talking about these proteins, you should always understand that they actually have 5 main domains. The real protein had 5 main domains. This is domain 1, this is domain 2, this is domain 3, this is domain 4, and this was domain 5. There are 5 domains. Now listen. Now listen with all your attention. These 5 domains are not numbered like this, but they are numbered like something else. Now listen. Their numbering has a peculiar type of arrangement, that is, it is known as, the first one is known as BH4 domain, second one is known as BH3, and the third one is known as BH2, fourth one is known by the name of BH1, and this is TM domain, that is transmembrane domain. That is, these proteins, are present in on the cell, and suppose this is a mitochondria, suppose this is a plasma membrane, and these proteins are present through and through the membrane. They are transmembrane protein, and this actually is the portion which is present here. Okay. Now, what I have, what is the name of this structure? What is the name of this mRNA, or what is the product of this mRNA? From this mRNA, what protein product I will get? The protein product from this type of structure is known by the name of anti-apoptotic proteins, or BCL2 or BCLXL. Okay, this is BCL2 or BCLXL. Okay. Now that means in BCLXL all the 4 domains, all the basic 4 domains are present. Page 4321. Now listen. Whenever I am talking about pro-apoptotic proteins, so that means whenever I am talking about, so this is what? This is the sequence of anti-apoptotic proteins. Anti-apoptotic proteins, right? Now these two are actually pro-apoptotic proteins. These two are the structure of pro-apoptotic genes. Now listen. But we know one thing, that under pro-apoptotic proteins there are 2 classes, right? Those which will form the channel, and those which will inhibit anti-apoptotic proteins, right? Now which one is which? Now listen. Let me draw the structures first, then you will understand. So this is the TM domain. TM domain will remain same, whatever be the protein. Okay, this is TM domain. So in all the three proteins, TM domain is present. And now in now you should understand one thing, that is, in this proapoptotic protein or in this proapoptotic gene, BH4 domain is absent. BH4 domain was there, but it is now absent. Okay. So what is the name of this protein, or what is the class of this protein? Now listen, these proteins actually forms channel in mitochondria. Okay, these type of proteins form channel in mitochondria. So what remains is this. Now listen with all your attention. In the last protein, you should always understand that is BH4 domain is absent, BH2 domain is absent, and BH1 domain is absent. Okay, and this gene, when transcribed and then translated, will forms proteins which inhibit anti-apoptotic proteins. Okay. So these are the genetic constitution of these proteins. Okay, now we will be talking in great details about the intrinsic pathway. Okay. Now we should talk about the intrinsic pathway now. Okay. Now listen. Suppose this is a cell, and suppose it is under stress. It is under stress, and it cannot take the stress anymore, and so this cell has this this old cell has decided to undergo apoptosis. Now listen with all your attention. From where we should start the intrinsic pathway? From where we should start? Now listen with all your attention. It's very important for everyone to understand that intrinsic pathway never starts actually from APOF1 formation or something like that, but we should always remember it always starts from a stress or an external environment which is intrinsically controlled, and it always starts from an external factor, but this external factor according to the cell is a very much disturbance. That's why the cell decides to undergo apoptosis. Now listen. When this type of disturbances occur, suppose according to a cell, what is the stress? A cell doesn't have any husband or wife like relation or something like that, but according to a cell, what is the stress? It can be heat, and one of the best stress is UV light. It can be UV light. Okay. Now listen. suppose this is mitochondria.